Hemoglobin. Hemoglobin definition is the iron-containing oxygen transport metalloprotein in the red blood cells of all vertebrates. Is the iron-containing oxygen transport metalloprotein in the red blood cells of all vertebrates. Genetics. Hemoglobin consists mostly of protein subunits, the globin molecules, and these proteins, in turn, are folded chains of a large number of different amino acids called polypeptides. The amino acid sequence of any polypeptide created by a cell is in turn determined by the stretches of DNA called genes. In all proteins, it is the amino acid sequence that determines the protein's chemical properties and function.
I've talked a lot about the importance of hemoglobin in our red blood cells, so I thought I would dedicate an entire video to hemoglobin. One, because it's important, but also it explains a lot about how the hemoglobin, or the red blood cells, depending on what level you want to operate, know, and I have to use know in quotes, these aren't sentient beings, but how do they know when to pick up the oxygen and when to drop off the oxygen? So this right here, this is actually a picture of a hemoglobin protein right there. This is hemoglobin. It's made up of 4 amino acid chains. That's one of them, those are the other 2. We're not going to go into the detail of that, but these look like little curly ribbons. If you imagine them, they're a bunch of molecules and amino acids, and then they're curled around like that. So this, on some level, describes its shape. And then in each of those groups, or in each of those chains, you have a heme group. You have a heme group here in green. Heme group. That's where you get the heme in hemoglobin from. You have 4 heme groups, and the globins are essentially describing the rest of it, the protein structures, the 4 peptide chains. Now this heme group, this is pretty interesting. It actually is a porphyrin structure, and if you watch the video on chlorophyll, you'd remember a porphyrin structure. But at the very center of it, in chlorophyll, we had a magnesium ion. But at the very center of hemoglobin, we have an iron. We have an iron ion. Iron ion. And this is where the oxygen binds. So on this hemoglobin, you have 4 major binding sites for oxygen. You have right there, maybe right there, a little bit behind, right there, and right there. Now, why is hemoglobin? Well, oxygen will bind very well here. But hemoglobin has several properties that, one, make it really good at binding oxygen, and then also really good at dumping oxygen when it needs to dump oxygen. So it exhibits something called cooperative binding. Cooperative binding. And this is just the principle that once it binds to one oxygen molecule, so let's say one oxygen molecule binds right there, it changes the shape in such a way that the other sites are more likely to bind oxygen. So it just makes it, one binding makes the other bindings more likely. Makes other bindings more likely. Now you say, okay, that's fine. That makes it a very good oxygen acceptor when it's traveling through the pulmonary capillaries, and oxygen is diffusing from the alveoli. That makes it really good at picking up the oxygen. But how does it know when to dump the oxygen? This is an interesting question. When to dump oxygen. It doesn't have eyes or some type of GPS system that says, oh, you know what? This guy is running right now, and so he's generating a lot of carbon dioxide right now in these capillaries, and he needs a lot of oxygen in these capillaries surrounding his quadriceps. I need to deliver oxygen. It doesn't know it's in the quadriceps. How does the hemoglobin know to let go of the oxygen there? And that's a byproduct of what we call allosteric inhibition, which is a very fancy word, but the concept's actually pretty straightforward. Allosteric inhibition. And when you talk about allosteric anything, it's often used in the context of enzymes. You're talking about the idea that things bind to other parts. Allo means other. So you're binding to other parts of the protein or the enzyme, and and enzymes are just proteins, and it affects the ability of the protein or the enzyme to do what it normally does. So hemoglobin is allosterically inhibited by carbon dioxide, and by protons. So carbon dioxide can bond to other parts of the hemoglobin. I don't know the exact spots. And so can protons. So remember, acidity just means a high concentration of protons. So if you're in an acidic environment, protons can bond. Maybe I'll do the protons in this pink color. Protons, which are just hydrogen without electrons, right? Protons can bond to certain parts of our protein, and it makes it harder for them to hold on to the oxygen. So when you're in the presence of a lot of carbon dioxide or an acidic environment, this thing is going to let go of its oxygen. And it just happens to be that that's a really good time to let go of your oxygen. Let's go back to this guy running. He's running. There's a lot of activity in these cells right here in his quadriceps. They're releasing a lot of carbon dioxide into the capillaries, well, at that point they're going from arteries into veins, and they need a lot of oxygen, which is a great time for the hemoglobin to dump their oxygen. So it's really good that hemoglobin is allosterically inhibited by carbon dioxide. Carbon dioxide joins on certain parts of it. It starts letting go of its oxygen. That's exactly where in the body the oxygen is needed. Now you're saying, wait, what about this acidic environment? How does this come into play? Well, it turns out that most of the carbon dioxide is actually dissociated. It actually dissociates. It does go into the plasma, but it actually gets turned into carbonic acid. So I'll just write a little formula right here. So if you have some CO2 and you mix it with the water, I mean, most of our blood, the plasma, it's water. So you take some carbon dioxide, you mix it with water, and you have it in the presence of an enzyme, and this enzyme exists in red blood cells. It's called carbonic anhydrase. Carbonic anhydrase. A reaction will occur that'll essentially you'll end up with carbonic acid. You'll end up with, we have H2CO3, and it's all balanced. We have 3 oxygens, 2 hydrogens, 1 carbon. And it's called carbonic acid because it gives away hydrogen protons very easily. Acids dissociate into their conjugate base and hydrogen protons very easily. So carbonic acid dissociates very easily. It's an acid, although I'll write in some type of an equilibrium right there. If any of this notation really confuses you, if you want more detail on it, watch some of the chemistry videos on acid dissociation and equilibrium reactions and all of that. But it essentially can give away one of these hydrogens. So you're left with, but just the proton, and it keeps the electron of that hydrogen. So you're left with a hydrogen proton plus, well, you gave away one of the hydrogens, so you just have 1 hydrogen. This is actually a bicarbonate ion. So this is bicarbonate right there, but it only gave away the proton, kept the electron, so you have a minus sign. So it's all of the charge adds up to neutral, and that's neutral over there. So if I'm in a capillary of the leg, let me see if I can draw this. So let's say I'm in the capillary of my leg. Let me do a neutral color. So this is a capillary of my leg. I've zoomed in just one part of the capillary. It's always branching off. And over here I have a bunch of muscle cells right here that are generating a lot of carbon dioxide. They're generating a lot of carbon dioxide, and they need oxygen. Well, what's going to happen? Well, I have my red blood cells flowing along. And it's actually interesting. Red blood cells, the capillary are 25%, their diameter is 25% larger than the smallest capillaries. So essentially they get squeezed as they go through the small capillaries, which a lot of people believe helps them release their contents. So releases maybe some of the oxygen that they have in them. So you have a red blood cell that's coming in here. It's being squeezed through this capillary right here. It has a bunch of hemoglobin. And when I say a bunch, you might as well know right now, each red blood cell has 270 million hemoglobin proteins. 270 million. And if you total up the hemoglobin in the entire body, it's huge because we have, let me write this number down. We have 20 to 30 trillion red blood cells. We have 20 to 30 trillion red blood cells, and each of those 20 to 30 trillion red blood cells have 270 million hemoglobin proteins in them. So we have a lot of hemoglobin. So anyway, that was a little bit of a... So actually, red blood cells make up roughly 25% of all of the cells in our body. We have about 100 trillion or a little bit more, give or take. I've never sat down and counted them. But anyway, we have 270 million hemoglobin particles or proteins in each red blood cell. Explains why the red blood cells had to shed their nucleuses to make space for all of those hemoglobins. They're carrying oxygen. So right here we're dealing with, this is an artery, right? It's coming from the heart. The red blood cell is going in that direction. And then it's going to shed its oxygen, and then it's going to become a vein. Now, what's going to happen is you have this carbon dioxide. You have a high concentration of carbon dioxide in the muscle cell. And eventually, just by a diffusion gradient, ends up, let me do that same color, ends up in the blood plasma. Just like that. And some of it can make its way across the membrane into the actual red blood cell. In the red blood cell, you have this carbonic anhydrase, which makes the carbon dioxide dissociate into, or essentially become carbonic acid, which then can release protons. So let me do the protons out here. Well, those protons, we just learned, can allosterically inhibit the uptake of oxygen by hemoglobin. So those protons start bonding to different parts, and even the carbon dioxide that hasn't been reacted with, that can also allosterically inhibit the hemoglobin, so it also bonds to other parts. And that changes the shape of the hemoglobin protein just enough that it can't hold on to its oxygens that well, and it starts letting go. And just as we said we had cooperative binding, the more oxygens you have on, the better it is at accepting more. The opposite happens. When you start letting go of oxygen, it becomes harder to retain the other ones. So then all of the oxygens let go. So this is, at least in my mind, it's a brilliant mechanism, because the oxygen gets let go just where it
Synthesis. Hemoglobin Hb is synthesized in a complex series of steps. The heme part is synthesized in a series of steps in the mitochondria and the cytosol of immature red blood cells, while the globin protein parts are synthesized by ribosomes in the cytosol.
All cells need oxygen. It is the essential fuel which is necessary to enable cells to stay alive and to carry out their various activities. Bringing oxygen to the cells requires the uptake of oxygen from the air in the lungs, its transportation in the blood, and its delivery to cells all over the body. The first step is the taking up of oxygen by blood flowing through fine capillaries in the walls of the lungs' air sacs, or alveoli. The oxygen molecules change from their state as a gas freely circulating in the air, dissolving into a solution in the plasma within the capillaries of the alveoli. Once in the solution of the blood, 98% of this dissolved oxygen is taken up by passing red cells, leaving just 2% remaining in the physical solution unattached. Red cells are particularly well suited to transporting oxygen because they contain a special oxygen-binding protein known as hemoglobin. Each molecule of hemoglobin itself contains 4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish blue in color and is called deoxyhemoglobin. It is heme which makes it possible for the red cells to pick up oxygen dissolved in the blood, transport it combined with hemoglobin, and release it back into the blood as oxygen in solution, ready for delivery to the various cells of the body. Hemoglobin gives up its oxygen as red cells travel through capillaries in tissues where there is a low content or partial pressure of oxygen. The partial pressure of oxygen represents the level of dissolved oxygen in plasma. As oxygen is released and again is carried in solution, the partial pressure of oxygen in the capillaries becomes greater than the partial pressure of oxygen in the surrounding tissues. This causes oxygen to move out of the capillaries into the tissues and to finally reach the cells. This graph, the oxygen dissociation curve of hemoglobin, shows why hemoglobin is particularly suited to its role in transporting oxygen. The oxygen dissociation curve demonstrates the relationship between the oxygen carried in combination with hemoglobin, the O2 saturation, and the partial pressure of oxygen in the arterial blood. The sharp upstroke and the flat plateau illustrate how oxygen is released to the tissues over a wide range of conditions. Its shape means that although the partial pressure of oxygen in the blood returning from the lungs and being pumped out by the arteries may be reduced to only 50% of the normal value, say due to lung disease or high altitude, hemoglobin will still be 85% saturated with oxygen.
Structure. Hemoglobin has a quaternary structure characteristic of many multi-subunit globular proteins. Most of the amino acids in hemoglobin form alpha helices, connected by short non-helical segments. Hydrogen bonds stabilize the helical sections inside this protein, causing attractions within the molecule, folding each polypeptide chain into a specific shape. Hemoglobin's quaternary structure comes from its 4 subunits in roughly a tetrahedral arrangement. Oxygen saturation. In general, hemoglobin can be saturated with oxygen molecules oxyhemoglobin, or desaturated with oxygen molecules deoxyhemoglobin.
Let's talk about exactly how oxygen and carbon dioxide come into and out of the lungs. So you know this is our alveolus in the lungs. This is the last little chamber of air where the lungs are going to interface with blood vessels, right? So this is our blood vessel down here. And oxygen is going to make its way from this alveolus. It's going to go into the blood vessel. And it's going to go from the blood vessel into a little red blood cell. This is my red blood cell here. He's headed out for the first delivery of oxygen that day, and he's going to pick up some oxygen, and it's going to get inside of the red blood cell through diffusion. That's how it gets inside. So the oxygen has made its way into the red blood cell, and where do you think it goes first? Well, this red blood cell is, we sometimes think of it as a bag of hemoglobin, right? It's got millions and millions and millions of hemoglobin proteins. So this is our hemoglobin protein. It's got 4 parts to it, and each part can bind an oxygen. So hemoglobin, I can shorten this to HB. Now oxygen is going to bump into, quite literally bump into one of these hemoglobins, and it's going to bind, let's say, right here. And initially it's kind of tricky because oxygen doesn't feel very comfortable sitting on the hemoglobin or binding to hemoglobin. But once a single oxygen is bound, a second one will come and bind as well. And then a third will find it much easier, because what's happening is that as each oxygen binds, it actually changes the conformation or shape of hemoglobin, and so each subsequent oxygen has an easier time binding, and we call that cooperativity. It has the word almost like cooperation in it. And kind of an easy way to think of cooperativity, the way I think of it, is that if you're at a dinner party, you're much more likely to sit where 2 or 3 of your friends are already sitting, if you think of this as a table with 4 chairs, rather than just sitting at a table by yourself, being the first one to sit there. So we kind of like sitting with our friends, and oxygen is kind of a friendly molecule, and so it also likes to sit where or bind where other oxygens have already bound. What are the 2 then major ways, based on this diagram, how I've drawn it, what are the 2 major ways that oxygen is going to be transported in the blood? One is hemoglobin binding oxygen, and we call that HbO2. just Hb for hemoglobin, O2 for oxygen. And this molecule, or this enzyme, then is not really called hemoglobin anymore. Technically, it's called oxyhemoglobin. That's the name for it. And another way that you can actually transport oxygen around is that some of this oxygen, actually I underlined it there, is dissolved, O2 is dissolved in plasma. So some of the oxygen actually just gets dissolved right into the plasma, and that's how it gets moved around. Now, the vast majority of it is actually going to be moved through binding to hemoglobin. So just a little bit is dissolved in the plasma. The majority is bound to hemoglobin. So this red blood cell goes off to do its delivery. Let's say it's delivering some oxygen out here. And there is a tissue cell. It, of course, doesn't know where it's going to go that day, but it's going to go wherever its blood flow takes it. So let's say it takes a path over to this thigh cell in your, let's say, upper thigh. So this thigh cell has been making CO2. And remember, sometimes we think of CO2 as being made, only when the muscle's been working. But you could be napping, you could be doing whatever, and this CO2 is still being made because cellular respiration is always kind of happening. So this red blood cell has moved into the capillary right by this thigh cell. And so you've got a situation like this where now some of the CO2 is going to diffuse into the red blood cell like that. And what happens once it gets down there? So let me draw out now a large version of the red blood cell, just so you get a closer view of what's going on. And we're in the thigh, and the two big conditions in the thigh that we have to keep in mind, one is that you have a high amount of CO2, or partial pressure of CO2, and this is dissolved in the blood. And the other is that you have a low amount of oxygen, not too much oxygen in those tissues. So let's focus on that second point. If there's not too much oxygen in the tissues, and we know that the hemoglobin is kind of constantly bumping into oxygen molecules and binding them, and they fall off and new ones bind, so it's kind of a dynamic process. Now, when there's not too much oxygen around, these oxygen molecules are going to fall off, as they kind of always do in a dynamic situation, except new ones are not going to bind because there's so little oxygen around in the area that less and less oxygen is free and is available to bump into the hemoglobin and bind to it. So you're going to literally start getting some oxygen that kind of falls off the hemoglobin simply because the partial pressure of oxygen is low. So one reason for oxygen to come into the cells is going to be a low PO2. That's one reason. So these are reasons, and I'm going to give you another one. That's why I'm writing reasons for O2 delivery. So one of them is going to be simply not having too much oxygen in that area. A second reason has to do with CO2 itself. So let's actually follow what happens once CO2 starts getting into the red blood cell. Now, this first CO2 molecule, it's going to meet up with a little water. Remember, there's a lot of water in the red blood cell. In fact, there's water all over the blood. In fact, it's made of mostly water. And so it's not too hard to imagine that a water molecule might bump into the CO2. And there's an enzyme called carbonic anhydrase. And what it does is that it kind of combines the water and the CO2 into what we call H2CO3, or carbonic acid. Now, if it's an acid, try to keep in mind what acids do. Acids are going to kick off a proton. So this becomes HCO3 minus, and it kicks off a proton. And notice that now you've got bicarb and proton on this side, and this bicarb is actually going to just make its way outside. So the bicarb kind of goes outside the cell. And the proton, what it does is it meets up with one of these oxyhemoglobins. It kind of finds an oxyhemoglobin. Remember, there are millions of them around. And it literally binds to hemoglobin, and it boots off the oxygen. So it binds to hemoglobin, and oxygen falls away. So this is interesting because now this is a second reason for why oxygen gets delivered to the tissues. And that is that protons compete with oxygen for, what are they competing for? For binding with hemoglobin. So they're competing for hemoglobin. Now, I said there's another thing that happens to the carbon dioxide. So what's the other thing? Turns out that carbon dioxide actually sometimes independently seeks out oxyhemoglobin. Remember, again, there are millions of them. So it'll find one, and it'll do the same thing. It'll say, well, hey, hemoglobin, why don't you just come bind with me and get rid of that oxygen? So it also competes with oxygen. So you've got some competition from protons, some competition from carbon dioxide, and when carbon dioxide actually binds, interesting thing is that it makes a proton. So guess what happens? That proton can go and compete again by itself. It can compete with oxyhemoglobin and try to kick off another one, kick off another oxygen. So this system is really interesting because now you've got a few reasons why you have oxygen delivery. You've got protons competing, you've got now CO2 competing with oxygen. So you've got a couple of sources of competition, and you've got, of course, just simply the fact that there's just not too much oxygen around. So these are reasons for oxygen delivery. So at this point you've got oxygen that's delivered to the cells, and these hemoglobin molecules, they're still in our cell, of course, inside of a red blood cell, and these hemoglobin molecules have now been bound by different things. So they're no longer bound by oxygen, so you can't really call them oxyhemoglobin anymore. Instead, they have protons on them, like this, and they might have some COO minus on them, so they might have, actually let me do that in the original kind of orangey color. So they basically have different kind of things binding to them, and as a result, the oxygen is now gone, and our system so far looks good. But let me actually now turn it around, and let's ask the question, how do we carry carbon dioxide from the thigh back to the lung? Let me start out by actually replacing the word thigh with lung. So now our blood has traveled back to the lung, and the question is, how much carbon dioxide did it bring with it, and in what different forms did that carbon dioxide come? So we've got a couple of situations. We've got a high amount of oxygen here, and we've got a low amount of CO2. So really quite different than what was happening in the thigh. So when the blood is leaving the thigh headed back to the lung, what's it got with it? Well, it's got a few things. One is that it's got hemoglobin that is bound to carbon dioxide, right? And this is actually called carbamino hemoglobin. And then it's also got some protons that are bound to hemoglobin. So the protons themselves are attached to hemoglobin. And just keep in mind that for every proton that's attached to hemoglobin, you've also got a bicarb dissolved in the plasma, right? Because it's a 1-to-1 ratio of these things. So you've got a bunch of bicarb in the plasma as well. And I'm writing in parentheses just so we don't forget that point. And finally, what else is in the blood? We've got some CO2 that just gets dissolved right into the plasma. So this is sounding a little bit like what happened with the oxygen situation where you had some CO2 in the plasma itself. And this is what's headed back from the thigh to the lung. So now in the lung, what happens? You've got all this stuff with you, and the first thing that happens is that you've got a lot of oxygen now in the area, right? A lot of oxygen in the tissue of the lung, and it diffuses into the cell, goes into the cell, and the oxygen is, because there's so much of it, it's going to go and try to sit in these hemoglobins. It's going to try to find its spot.
Oxyhemoglobin is formed during physiological respiration when oxygen binds to the hema component of the protein hemoglobin in red blood cells. This process occurs in the pulmonary capillaries adjacent to the alveoli of the lungs. The oxygen then travels through the bloodstream to be dropped off at cells where it is utilized as a terminal electron acceptor in the production of ATP by the process of oxidative phosphorylation. It does not, however, help to counteract a decrease in blood pH. Ventilation, or breathing, may reverse this condition by removal of carbon dioxide, thus causing a shift up in pH. deoxygenated hemoglobin. Deoxygenated hemoglobin is the form of hemoglobin without the bound oxygen. The absorption spectra of oxyhemoglobin and deoxyhemoglobin differ. Deoxyhemoglobin has significantly lower absorption of the 660 nm wavelength than deoxyhemoglobin, while at 940 nm its absorption is slightly higher.
The transport of oxygen and carbon dioxide between the lungs and tissue cells is the prime function of red blood cells. Essentially, red cells are packets of hemoglobin, a molecule which readily combines with oxygen. As the red cells flow through the body tissues, oxygen diffuses from them to the tissues in exchange for carbon dioxide. The deoxygenated red cells carry a little carbon dioxide, then pass through the bloodstream to the lungs. Here the reverse exchange of oxygen and carbon dioxide takes place. The oxygenated red cells are then conducted back to the body tissues.
Iron S oxidation state in oxyhemoglobin. Assigning oxygenated hemoglobin S oxidation state is difficult because oxyhemoglobin HbO2 by experimental measurement is diamagnetic, no net and paired electrons. Yet the low energy electron configurations in both oxygen and iron are paramagnetic.
Hello and welcome to another episode of Interactive Biology TV, where we're making biology fun. My name is Leslie Samuel, and in this episode 60, I'm going to be talking about hemoglobin and what's called the oxygen dissociation curve. So let's get right into it. We've already done an introduction to the respiratory system, and we've shown how the heart beats and sends the blood. When the right ventricle sends the blood, it sends it to the lungs. That comes back to the left atria, and then the left ventricle pumps, and that sends the blood through the rest of the body. We've also looked in the lungs and seen how we have the trachea going into the bronchi, and then that splits off into the bronchioles. And as you can see here, that gives us the alveoli, and it's in the alveoli where we have the exchange between the oxygen coming into the bloodstream via the capillaries that we have here, and the carbon dioxide leaving the capillaries, going into the lungs and being sent from the body. Now, when the blood comes in here, it is picking up oxygen. And the type of blood cells that are picking up the oxygen would be the red blood cells. And here you can see a picture of a few red blood cells. Of course, it's simplified. It's not showing the white blood cells or anything else. It's just showing the red blood cells. And these are the blood cells that pick up that oxygen. Now, in the red blood cells, we have a special molecule, and that molecule is called hemoglobin. And you can see a three-dimensional image here of the structure of hemoglobin. So this is, let me write it here, hemoglobin. And this molecule, it's actually a protein, and this protein is the protein that is responsible for picking up the oxygen. Now let's go into a little more detail. You can see here that we have these four structures, and those four structures are called, let's do that in blue, those are heme groups. All right, so these are the four heme groups. The special thing about these heme groups is that those are the parts where the oxygen is attracted. So we have O2 that actually comes and binds to the heme groups. As you would imagine, since we have four heme groups, we can take a total of four oxygen molecules. So this is one oxygen molecule here, and we can have another oxygen molecule here, here, and also here. So this hemoglobin molecule, once again, has a capacity to hold four oxygen molecules. Now, what's interesting about the hemoglobin is that whenever one oxygen binds to a heme group, that causes the entire hemoglobin structure to undergo a conformational change. So we're basically changing the shape of the molecule whenever one oxygen binds. Now, as you can see here, this is the heme group, but there's stuff around it, and you can imagine that it would be relatively hard for the oxygen to get in there and find the right spot. However, when one binds, it causes a change which opens it up a little bit to make it a little easier for another oxygen to come in and bind. And when that other oxygen comes in and bind, it causes another conformational change, making it easier for another oxygen to come and bind. And once again, once that oxygen comes and binds here, it makes it easier for another oxygen to come in here and bind to it. So in other words, as it starts taking up oxygen, it makes it easier for it to take up more oxygen. And then of course the opposite will be true. If we have a hemoglobin molecule that has four oxygens attached, and for some reason it gives up one oxygen, that's going to cause a change that makes it a little harder for the other oxygens to bind. In other words, it becomes easier for oxygens to leave. So as oxygen leaves, it's easier for more to leave. As oxygen binds, it makes it easier for more oxygen to bind. And as a result of this, we get a relationship that is shown in this oxygen dissociation curve. And what you can see here is we're going to be looking at this blue line. And as you can see here, it's not a linear relationship. In other words, as the amount of oxygen increases, so here we're showing the pressure of oxygen, as the pressure of oxygen increases in the environment that the hemoglobin is in, you're going to get more binding, making it easier for more to bind, making it easier for more to bind, and the graph is going to increase faster as you're going to the right, where you have an increased partial pressure of oxygen. Not a linear relationship, but as some binds, it becomes easier, so more bind faster. And it becomes easier and it gets faster and faster until, of course, it reaches to where it's getting saturated and it dies off. Now, you might be wondering why it's not just 4 levels, since we only have 4 binding spots for the oxygen, the 4 heme groups. However, this is not looking at one hemoglobin molecule. This is looking at a bunch of hemoglobin molecules in a bunch of red blood cells. And overall, as some start binding, it makes it easier and easier, so it's going to increase faster and faster until it reaches to the saturation, and then it's going to slow down when it reaches its full saturation. Also, as you come in this direction, as the pressure of oxygen decreases and oxygen starts to leave, Here it's leaving slowly, but as they start to leave more, it's dropping down faster and faster and faster until all of the oxygen is gone. So this is the oxygen dissociation curve, showing once again, as you pick up oxygen, it makes it easier for oxygen to be picked up. So here it starts slow and it goes faster and faster and faster and faster. And as you release oxygen, it makes it easier for oxygen to leave, and then that goes down faster and faster. This is called the oxygen dissociation curve. That's pretty much it for this video. If you want to see more videos like this and check out the other resources we have available, visit the website at www.interactive-biology.com. That's it for this video, and I'll see you in the next one.